So we're going to go ahead and get started. I'm Leanne West, for those I have not met, though I think I have met probably everybody in the room just about. I am helping with GTRI interact with the academic side, looking at the intersection of biology and engineering, kind of thinking about what are the next big problems on the horizon in the biology, biosciences landscape, and what we can do from there if we partner with GTRI to maybe build things that can be used in the real world. So that's what we're trying to do. We want to think big, like an ARPA-H program or a DARPA program or a BARDA program, something like that, right? So what would be a game changer out there that we could do? And so what we're doing is we're hosting a series of workshops to allow people to hear from each other about what type of research is going on, to hopefully spark some interests and thoughts on ways we can collaborate. And there is a funding mechanism to go after. It's going to be due in the new year, but we have information on it now. So you can go ahead and start thinking about that and putting teams together. And we are here to help you do that as well. And I'm actually very excited that we have a bunch of our IBE steering committee members. If you just want to raise your hand for a second. There you go, Rebecca, Cindy, Nick, Brian, Greg. Yeah. So, so I'm glad everybody's here. So this is the team you can come to for more information or advice, or you can come to me or Trina, and we can put you together with people. So we're going to go ahead and get started. And our first speaker, I think, is Ed Botchway. If you want to come on up, and this is your microphone that they can hear, and this is the remote. I think if you need it.
Apologies, if you'll just give me a second to get my laptop hooked up.
Give me a second.
Actually, am I gonna need to share into the Zoom?
Oh, you probably will. And Trina just walked out of the room.
Okay, I need the Zoom link. Apologies, I just realized.
Yep. Let me send it to you right now. What is is it Edward?
Yes. My last name is more distinctive. Yeah, there you go.
Conflict because you're here, but you should have it now on your board if you don't.
On my
Your calendar, I would think.
This I made this, so it doesn't have a thing. If hold on, there might be a brute force method.
Okay. So how about while we do this, if you're not on the steering committee, maybe we can be introduced to you. So just to know who's in the room with us, would that be good? I know you're going to be presenting here in a minute, but do you want to introduce yourself?
Yeah. Hi, my name is Vladimi Tsukruk. I am Regional Professor in the Geoscience Engineering Department. Do a lot of biology, engineering, materials aspects, mostly in collaboration with Air Force Research Lab and also and treatment.
Wonderful. All right, back row.
I'm Al. I was emailed this workshop and thought I'd show up in the bioengineering program, a third year PhD. Yeah,
Awesome.
I'm excited to hear. Oh,
Awesome. Right.
Hi, my name is Parma Haji. I'm a student assistant in the ACL with the AWTP. I'm with a bio cyber security project. It's very interesting. very interdisciplinary, so I thought I'd show up.
Trying to get the
He's trying to make his
session
presentation.
ID or do I need to, I think. Ooh, okay.
Or it might be too big, but can we just get the,
The Zoom- If I can get the Zoom link, I can. Oh,
okay.
Yeah, yeah. I'm Farzaneh and Najafi. I'm assistant professor at Biological Sciences.
oh, is it team?
I do these systems and behavioral and patient neuroscience. So we start with them on the basis of behavior. And see how they go with the patient. And, yeah. That's
Great.
I'm Tabitha Rosenbaum. I continue to report. I'm an ACL and LD. I have interest in human performance. My background is electrical and biomedical engineering. I have a lot of interest in the neurospace, but also in alternate spaces that overlap between EE and IE.
All right. Person who just walked in the room. Do you want to introduce yourself? Just say hi, say what, with your first time.
I'm Valeria, I'm a postdoc in Vladimi, Tsukruk, working on material science and engineering.
Awesome. Okay.
I'm not getting it from anywhere. Oh.
I tried. Sorry, everyone online for the technical difficulties. We'll get this fixed in just a second.
Yeah, I see. Botchway, yeah. Getting. Okay, oh, you missed it.
Okay. Because I tried forwarding the invite and you didn't get it.
Then.
Is there a way we could hook up his computer to? But not to this, not to the meeting?
Getting.
Can we switch the order so we can do that, like switch the order of speakers and then get someone from IT and then we can figure out how to get his laptop connected?
Okay, do we have the other presentations? You're ready to go?
Okay,
Are you okay with that, Ed?
that's fine.
Okay,
Okay.
well, then, up next. I will take the.
Sorry.
All right, everyone. Sorry about the delay. We were having technical difficulties with the first presentation. So we're actually going to go to the second presentation. So Farzaneh, if you want to come on up, we're going to be talking about systems neuroscience and predictive processing. So they can hear online. You can put it on yourself, put it in a pocket. Yeah.
Okay.
Yes. for that.
Thank you.
There's six.
Okay, hi everyone. I did already a brief introduction. I'm Farzaneh Najafi. I'm a relatively new assistant professor at the School of Biological Sciences. I started January 2023. And I'm going to talk very broadly about the type of research we do in my lab. The question is about closer, sure.
Yeah, thank you so much.
How is it better?
You can probably flip it to.
Yeah, let me just flip it. Is it better? Okay. So the question is about how the brain makes predictions about the world. That picture I've chosen in the study with perceptual and motor behaviors, and kind of is showing a complex motor behavior here. We do there is laser. I think I should just not click on this without. Is there a laser pointer?
Get this to play. Okay. Okay. So you're.
Oh yeah, I have it.
Okay.
Thanks. Thanks. And then this advances.
Advance.
Got it. Okay.
There you go.
Awesome.
That's me.
Sure.
Here.
I can also just use the.
I don't know why that thing was working 2 seconds. So. There we
Okay,
go.
don't worry. So this is a picture of the neurons that we image in the brain. This is the computational type of analysis that we do. I will talk about this. So what we study is what are the neuronal circuits and computations that underlie predictive processing in the brain. What we mean by predictive processing is how the brain makes predictions about the world. So what that means is that our brains are not just sensory processing units. They also actively anticipate the world. And then there is this actual sensory input, and then there is this prediction of the world, and these two get compared. And that's how we learn. If there is a mismatch, then we learn to update our predictions. The process of learning is really this process of updating predictions about the world. So I hope this concept of predictive processing and what that means for the brain is clear to everyone, regardless of their needs. So sometimes we refer to this as an internal model of the world because there is this external world and then we have this internal simulation of the world. And okay, which generates predictions. So
Yes.
Yeah, that's a good idea. So I'm going to do that.
okay, so we have an internal model that simulates the world, generates predictions. And then we have those external inputs that get compared. And then if there is a mismatch, the predictions are updated. So the besides this being a very interesting problem on its own, there is clinical relevance to this. One that a lot of people might relate to in different ways is that if the external world is disconnected from the internal world, meaning that if our internal model is not functioning properly, then that leads to psychotic symptoms. For instance, in schizophrenia, there are auditory hallucinations are very common. And one way to look at that is that the person's internal model is generating these incorrect predictions about the auditory inputs that in a healthy subject, they would get updated. And the healthy subject's brain would say, no, this is not what you are hearing, even though you thought, but it's not what's out there. But in a schizophrenic brain, this doesn't happen. Or in the case of psychedelic drugs, same concept, like with visual hallucinations. There is this detachment between the external world and the internal world. Similarly, in the motor domain, when the sensory motor environment changes, for instance, walking on a street versus walking on a train, we need to update our movement plans. And one way to look at that is that we update it because we have now different predictions about the sensory consequences of movements. When we walk on an even surface, we predict that this is going to be this level of hardness, the texture is like that, so I need to apply this much force when I'm walking. But when we go on a train, we need to update it. And if we cannot update our internal models of the motor, of the sensory motor world, then we'll encounter impairments in motor adaptation. So it's really a framework, a theoretical framework for looking at the function of the brain. To put it a bit in more concrete words, because this is about intersection of biology and engineering, that's what's happening. Internal model generates predictions of the next sensory motor state. Then there's this part of the brain that we think does the comparator job. It receives those predictions and compares them with the actual sensory input. If they mismatch, there is a prediction error. Now it goes to this part of the brain that learns. This is the plastic part of the brain. It learns and adaptation occurs, and then now we have an updated prediction. This updated prediction goes to this part of the brain, which is a controller. It generates percepts or movements. And then those lead to a sensory motor output, and that's how this loop is completed. And then the idea is that there's a copy of these movement commands that as they are generated, they go to the internal model, and that's how we can perform online adjustment of movement. So we don't wait for the feedback. As the movement is being generated, the brain receives a copy of it and makes predictions, and that's how we can online adjust movement. So the whole idea of this predictive processing is that it bypasses all the sensory feedback delays that exist in the system, and it generates these feed-forward predictions.
We have time for questions now, or should we?
You can ask one now.
Yeah, go ahead.
Okay, you might have to repeat it for online, but are there, knowing the brain can do many things somewhat in parallel, I'm not an expert on that, but are there cases in which this diagram, some things occur at the same time, like maybe the comparator goes to the controller and straight around as well as going to the adaptation sites? Like there's so many things.
Yes, I mean, this is a very simplified model. And what we're trying to do, for instance, one of the main objectives of my lab is to map the brain onto this model. The reality is that it's way more complex than what I'm showing here. Yes, the idea is that the brain needs, we think the brain is performing this computation. There are some key elements there. The element of the internal model, the element of the comparator, there is an element of the controller which generates. So there is the one that executes, there's the one that generates the prediction, the one that takes the prediction to execute the action, and the one that compares this action. That how are they connected, and whether I don't think it's like this. Yes, yeah, sure. Thanks for the question. So what we think in terms of this mapping is that the cerebral cortex, which is basically this more perceptual part of the brain, does this part of generating predictions and updating them. And then the cerebellum, which is the small brain, basically in the back of the brain, that's the one that performs this updating of the, not the updating, the comparison of the predictions with actual sensory inputs. And then the idea is that even though multiple anatomical pathways, so mouse's brain, cortex, cerebellum, multiple anatomical pathways in both directions connect these two regions. But the majority of current studies that have looked at internal models have either focused on the cortex or the cerebellum. There is really very little studies that have focused on the connection of the two, and that if we are saying that both of these regions have internal models of the world, then how is it that these two are different? Are they about different aspects of the world, or are they about the same aspect, but they use different computations? So why would the brain have this redundant way of predicting to these different regions? So those are the main questions that we are trying to address. There are at the moment three research directions. One is about movements. So we work on multiple behaviors in the lab. This is one that we have developed. It's like a joystick in front of the mouse, and the mouse pushes the joystick, and then we train them to do this in a very specific temporal window. And if they do it, then we reward them. So major questions here are how our brains generate temporal predictions about movements and how they update them after censoring all their errors. And an example of that is when we have to adjust our serve timing, for example, in volleyball or some other sports. So what we do is that we train mice for these, and then while they perform this very form, we both read and write their activities. And when I say read, I mean that we measure it whether with optical methods or electrophysiological methods, and we write it by manipulating it again optically or electrically or chemogenetically. The second direction of research in the lab is focused on perceptual behavior. So here mice see visual stimuli at different timing intervals, and they have to judge whether the temporal interval increases or decreases. So it's a decision-making type task. And you see there are multiple scouts in front of them that they report their choices because, of course, they cannot speak. So we train them that they report their choices by looking to either center, left, or right scouts. So the key question here is how our brains generate and update temporal predictions during perceptual tasks. And an example of that is when animals adjust their hunting strategies seasonally, right? They go to one place to dig and get food, and they see it doesn't exist, and they have to update that strategy depending on the time of the year. And then there is a third direction in the lab, which is about sleep. This one is not directly along the lines of perceptual decision making. This happened because I got this collaborative award. And the key question here is that sleep and how the brain sleeps has been addressed with cortical and hippocampal areas, but the cerebellum is completely left out. You might wonder why, because sleep is historically associated with lack of consciousness, and cortex is where we think consciousness happens. So that's why for the lack of consciousness, also they have focused on the cortex. But now there is more and more evidence for many years coming out that shows the cerebellum is involved also in cognition and perceptual tasks. One of the key things that happens is that in autistic children, when they study the cerebellum, there's massive degeneration of cerebellar neurons. So that's why we thought we can study the cerebellum for the sleep line. It's something that no one has studied. So I've heard with this very famous sleep lab, and in the lecture you got this grant. So the idea is to study the cerebellum during sleep, to study whether the cerebellum sleeps or not and how. Because one thing that many of us may not know is that the sleep is very local in the brain. As I'm speaking, there might be parts of my brain that are sleeping. So it's not that all of the brain necessarily sleeps. So we want to know if the cerebellum also sleeps. And if it sleeps, what's the functional significance of it? We're going to start with motor behavior. That again, this is another behavior we have developed in my lab. That it's a cerebellar dependent behavior that they adjust the amplitude of their eyelid closure depending on the strength of the aversive stimulus that they receive. So that's how it looks like. And I chose this behavior because this is an established cerebellar behavior. So I needed to be very conservative because the question is really cerebellum here. And then the idea is that we are now sleep depriving these mice. Right now that I'm speaking, actually, we're doing our second imaging session from these mice during sleep deprivation, during sleep and sleep recovery. And then we want to see how that affects the function of the, how sleep deprivation affects the function of the cerebellum and cerebellum dependent motor behavior.
That partial local sleep, is that just in the mice or is that in?
Humans, yeah, yes, yeah. And that's a very interesting. Okay, so these are now data. I'm not sure if you want. How much more time do I have? Because the conceptual part is now done, and I don't know if people are interested.
Maybe like 4 minutes or so.
4 minutes. Okay, sure. So if let me see, I have this. Okay, sure. So this is for those who are interested in the. Oh, sorry, I'm going in there. There should be some. Yeah. Okay, so I just showed things that might be interesting. Yeah, so these are just the details of the behavior and how we adjust the timing and how we make it such that mice make predictions. The way we do it is that we repeat the same, we require them to do the same thing, and then all of a sudden we change the task rules. So they have to figure it out and they have to update now their performance. So you might wonder, how do we study predictions in mice? That's really the trick that we use. Repeating, so they learn it, and then violating it without any cues, and then the mouse has to figure it out. And I guess there was one more. Yeah, and this is these are the trajectories of the movement that the mouse does. Like it's double press, and they have to time it. They do it visually guided or self-time, because we want to compare this how this predictive processing happens when I self-time their actions versus it's guided with an external cue. And then we show that there is a movement timing adaptation. And then here we have done the chemogenetics to basically manipulate individual areas. And this is part of the cerebellum. And when we manipulate the cerebellum, you see that the performance clearly drops. So they need the cerebellum to perform this movement timing adaptation. Then we have parietal cortex. Parietal cortex is more of a high-level area in the brain that generates high-level predictions about the world. And so what we see in this area, it doesn't affect the movement execution as much, but changing from one context to the other context or updating the predictions seems to be involved in that. We can see the drop. You may wonder how these things look when we record. This is the cerebellum, beautiful protein cell dendrites. They look like decorative trees. We are looking at them from a plane that shows from the top. You see, this is one folio, another folio. This is the parietal cortex. We have amazing inhibitory neurons in red, then the rest of the neurons in green. So we managed to simultaneously image them. This is how we do segmentation to identify the neurons. Then we extract the fluorescence traces, and then we do the analysis that we have on those. So these are two recordings from the cortex, parietal and the cerebral cortex. These mice are transgenic, but they express these fluorescent dyes in specific neurons, and that's how we image them. So these fluorescent dyes are basically sensitive to the concentration of calcium in the cell, so they're calcium sensors. And the reason we care about calcium is that calcium is directly related to the activity of neurons. So when neuronal activity changes, calcium concentration changes. So a calcium sensor helps us to have a proxy for measuring the activity of neurons. Then we measure things like how many neurons are sensor responsive, reward responsive, licking responsive, and such. And then we do analysis to look at basically the lower dimensional subspace in the activity of neurons to see how these trajectories evolve as the mouse performs an action. So this is when they do like fast tempo movements. This is when they do slow tempo movements. And then these are all the additional analysis that we do, decoding and such, to see what's the information what's the information and what's the lower dimensional subspace the activity of those.
The principal components in that lower dimensional subspace, what modality is that within? What
So
space?
we have 3 dimensions: neurons, time, and trial. Trial is like double press. This is one trial, another double press. This is another. So we first reduce it to neurons times time. So we take an average across a window that we think that's in, and then we reduce the dimension of neurons. So the idea is that we have 205 or 500 neurons. So what's the low-dimensional subspace in the activity of these neurons that represents the feature we are looking for? So that's it. But we can also do it for time. We can also reduce the dimension. So it depends on the question. And the ongoing work that we have is that we want to identify these prediction and prediction error subspaces. So for that, we're working with tensor component analysis, because now we need there I said we average it, but now we need the 3 dimensions here. So we want to identify the dimension, but for instance, prediction error gradually increases and predictions go up as well. And so on. And this is the line that we have now, that I was very much going to 3 graduates, to a PhD, multiple masters and such. Yep. Thanks.
Thank you.
Kind of special, so I'm imagining that you have a lot of animals in the animal facility here at Georgia Tech,
Yes.
and you must have some really special considerations for your animals, right? In terms of
Yes.
light dark cycle, temperature,
Yes.
feeding, changing.
Yes.
Do you guys like do that yourselves, or do you? I'm
So
just curious.
the main thing that we have is the reverse light cycle, because mice are nocturnal, so they are active when it is dark. So we want to run our behaviors when it's their night. So that's why we reverse their cycle. And the animal facility, so they have from 7 a.m. to 7 p.m., they do the reverse cycle, and we keep our mice in that room. And for the sleep project, there was a lot of considerations and back and forth with the animal facility with that, but we finally managed to do it because we have to sleep deprive them.
Yeah.
So they were careful with the health. Yeah.
Good.
Yeah.
Thanks.
All right. Thank you. We have the microphone.
Oh, I'm sorry.
This advances slide.
Okay, can you hear me?
Yes.
Mic is on? Okay, good. So let me go to first slide. Yeah, so I am Vladimir Tsukruk, which is a professor at Georgia Tech for a while, already 18 years, material science and engineering department. My lab, formal name, Service Engineering Molecular Assembly Lab. You did change this for 30 years, it's still valid name. So what we're trying to do is trying to see how you can build up different materials from molecular level for different applications. I'll talk briefly about what we're doing right now, Chirooptical biophotonics materials. We do a lot of different projects, mostly supported by Air Force and Department of Defense, DoD as well sometimes. So that's a summary of what we are doing, different materials. Polyanionic liquids is energy storage, trying to control ionic solid polyalkylite. Two-dimensional materials, we work a lot with graphene, nanomaxines, and different structure conducting materials for different potential applications. And bio-related things, we work with a lot of what our favorite is nanocellulose right now. We work a lot with the silks, different types of the silks as well. We don't do DNA, we tried, but it doesn't work good. And sensors, we do sensors, photonic combined with pressure sensors, discrete photonics materials, magneto-optical materials. And our specialty is high resolution AFM. We try to understand what's happening at molecular level with our material before we start building up something for different applications. So it's a summary. This is two slides from a recent review published in Responsive Materials. When you talk about what you can learn from nature, and in this case, a chirality and helicity, how it works, it's studied from DNA, molecular chiralities, and going to different types of the exoskeletons that create a structural color because of the presence of this chemical structure, periodicity with the different refractive indices. On the top of this, it's also additional mechanical properties. Then going to microscopic chirality for different potential applications. This is what we discussed in this particular review. And the next review, it's Nature Materials review. We talk about what can be done in terms of the toughness and strength of the materials. And this is summary what my students generated. We basically take a look at Young's models, strongest possible materials, take the toughest materials here. And you can find a lot of classical materials like minerals here, particularly ceramics, metal composites, fibers, particularly polymers and elastomers. And if you look what's happening with biological, was a special review focus on what you can learn from biology and how we can use these biological materials. So you can see, yes, chitosan here, silk here, graphene oxide with a paper cellulose, CNC cellulose nanocrystals with pairs. You can see that they are silk with graphene oxide. They're pretty competitive. They're competitive if you need stretching elastomeric type materials, but they're also competitive with cellulose nanofiber silks they're competitive with metals, materials, ceramics as well. So there's a room for bio-inspired and bio-derived materials. Depends upon what you need. Of course, we can compete with a temperature. If you need high temperature, it's still limited to 300-470 degrees. But in terms of the presence of the colors, photonics, or in terms of the mechanics, so we can compete with the different classical materials. So when I tell you a couple of things, what we are doing right now is very brief summary. Yes, we are doing so-called discrete materials for optical photonic applications and printed materials for different applications. What I told you recently, we moved a few years ago from silk to cellulose, different types of the cellulose materials. These are nanocrystals, nanocelluloses. So you can see it's a beautiful needle-like structure from nanocellulose. Typical length, couple hundred nanometers, typical diameter, 300 nanometers, and the elastic mode is about a couple hundred micropascals. Think about like steel, small steel needles, nanoscale needles, extremely strong. Yes. So you can make a lot of interesting composite materials. You can do chemical modifications. If you look along this needle, they look like this. It's basically like 5, 10 chains, cellulose chains, one direction, 20 chain, one direction. The beauty of this, there's combination of hydrophobic, hydrophilic faces, is creates so-called antiphilic nature. They can control themselves at interfaces. They form different interesting structures. And one of the interesting structures that we work with, it comes from chiral or helical structure. They don't pack it, they try to pack, they don't want to pack like normal isotopic structure. Those pack with twist, it's carotid, and when they're twisting from layer to layer, they form this pitch in the visible range, and so the structure comes highly reflective, highly iridescent, a broadband as well. So if you're working on these materials, trying to move to infrared for some applications, trying to make a very narrow band. This is my work with Air Force Research Lab. This is how they look like when you break them. It's basically it's a single pitch. So you can look at those, you can look at the structure, you can see that if you break it down, so you're pulling out pieces of this nanocrystals from this chiral structure. When you're working right now, if you work for a while studying mechanical properties, what you call discrete materials, you do 3D printing, and you see if this movie works. Okay. I'm doing that. Yeah, going back. It's supposed to be a movie to show the kind of move from continuous chiral classical crystals with interesting photonic properties to Bragg style, Bragg mirrors, or photonic band gaps. And when you print them like discrete chiral you can rotate, you do printing with twisting. So you can print in any type, or you can print left-handed, right-handed structure. You can control twisting angle, and you can create different discrete optical materials. This is what you're doing right now. We're into infrared for a lot of different reasons, including thermal isolation, including optical communication, and it's next to impossible to do with a regular liquid crystal materials. Yes, and natural materials as well. So all these natural nanocellulose, they form visible, the beautiful visible rays. They don't show anything in the infrared. But because now we deal with blocks, we're creating blocks, and then we can orient highly anisotropic blocks. We can orient them in different directions. This how we are doing right now. It's a layer by layer Dr. Blade printing. So it's scalable. We do a lot of different things, but this is our favorite right now. You're putting a droplet of your suspension, you can Dr. Blade, you can move, you can create highly oriented structure, and you can do shearing, you can do natural chemical structure inside of this. You can shear them in one direction, and then this kind of example where you build blocks. You repeat the steps multiple times, and you can create different types of materials, different properties. Of course, you look at the individual properties of these layers, you can see the needles. So the size of this spectroscopical absorption, you look at their refractive indices for photonics is very important, delta M as well. So how depends upon wavelengths. Look at the anisotropic, you study different types of biofrequencies to understand the individual layers and to understand what's happening when you build up light printed structure. This is example we just published a couple of months ago, the same printing process. Of course, there are some tricks involved when you build a composite. In this case, this will be CNC block, highly oriented. We have to kind of glue them together, printing, so to create robust composite materials, building out layers. we can control up to synthetical layers. We have to do at least a couple of pitches to be sure that photonic response is doable. And typical thickness of this layer, you can see we work with a 100 nanometer thickness layer and look at the physical properties. This will be next. It shows what we are doing, printing structure. This example would be measure CD, circle dichroism. And you can see that you print clockwise, counterclockwise, you can create left-handed, right-handed structure from the same materials. It's an artificial induced chiral structure. You can do emission as well. If you're getting dyes, you can do emission with a proper chirooptical proof. It's a circle polarization, very important for encryption, for example, and for different types of applications. The next example, this is what we are doing right now. You look at the classical break stacks with different anisotropy and doing twisting. In this case, the twisting happened at 45 degrees. It's kind of confusing because it's not clear. You have left-handed propagator line or right-handed propagator line. You can see we can create band gap structure, 1.4 micrometer approximately band gaps, and we basically create, in this case, dual chirality. So you have light propagating, you can have right-handed, right-handed at the same time, in addition to what normally you can have on the left or right or left-handed. So it's just start with video very recently. And a couple other things. I show you example very briefly. So you do biocomputing as well. It's a thin film transistors where you try to control multivariate logic, but I don't have time to talk about this. I'll show you just one example. If I can start this movie, let me see. Yeah. This is a recent walk we are doing collaboration with a Florida Robotic Institute. It's like university system of Florida, similar to GTRI, just 10 times smaller. Yes, and this is what we do. They're working with this robot Nadia. They're trying to make humanoid-like robots instead of like big Boston Dynamics robots. And what the problem with the Nadia is that there's no sensing, no feedback from environment. Yes, and they invited me a couple of years ago. We are doing our project. So basically, it's a pre-programmed structure, easy to work on the flat surfaces. But you put this robot, normal robot, in the Senti or Medi stuff, and they basically sync immediately. This is one example of what we are doing there. So they do like different uneven surface, and key point is to create sensors on leg of the Nadia on the arms as well at some point to be sure that they can open doors and they can walk there, basically sensing. Right now, these very simple piezo elements, you're trying to recreate a full sensitivity of the human feet with the arrays of the sensors, pressure sensors.
Is that Matt Johnson? Who's a collaborator at IHMC?
At IHMC, we have Morley Stone, he's director of this stuff, and also Johnson, the part of this, yeah. So working with Morley Stone there. So he was at FRL before, he moved there, he was CTO at the FRL Human Performance Division. Okay. Now he moved up to Ohio State, he moved there. So we work with them. And this example, what we are doing now, you can see, so sensors, current sensors are not very good. They're usually like boxes, metal boxes, or 3D gel electrodes they put on your skin, and you collect signal from your skin. What we are doing now, we're trying to create second skin, very, very thin layers. It can be conformally built in your body. You can see this is artificial finger because we study morphology. This is how it looks like on a human skin. This is what we look at conformality. You can see that basically our film is conductive tri-layer composite film. The sole thing that's going around all topography, very complicated topography. As a result of this, so you can cover this, you can collect very good signal. And I don't show this, it's unpublished and we're still getting the permission to work with a human subject. But we are doing this to show that you can do ECG and G signal collection from the people on emotion. This is the big stuff for military as well, because they want to monitor health and state and cognition of the people when not just sitting like this and making ECG, but running and doing a lot of different things. My postdocs degree spent almost in Florida, going outside 95 degrees, going to cold room and watching what's happening. Can really collect this after sweating several days and do this kind of stuff. This is my more applied oriented project right now with the Florida Research Institute, a couple of different projects for them. This is just summary of collaborations. Guys were involved in the studies. I told you supported by NSA referral. This is different structural color for cellulose finger. And this collaboration was more a team bonding and drinking about mostly at Pearl. And I'm Ukrainian American also. So we stand for Ukraine and then to thank you for your attention.
Anyone have questions?
Well, thank you, Professor. There are lots of interesting things in there, and I don't know which one to ask about first, but you knew I would have a question, Leanne. Thank you. So you mentioned a biotransistor, I think, in there somewhere along the way. Could you say a little more about that? And then you also mentioned piezoelectrics. I guess that's the transducer. How does the transistor play?
Yeah, we published several papers in my NSF project. What we are doing with we're doing film transistors right now with organic semiconductors. What is the problem? This is a standard logic, yes, 0-1 logic, normal tool. We're trying to create multi-level logic. When we adding up structural color cellulose, you have lasers here, and you're basically shining different lights. You create different response of semiconductor channels, and you can do not just 0-1, you can do 0, 1, 2, 3, 4. You can multi-level visual of 13 different steps. As a result, it's controlled by light. What's called biophotonic transistors. Basically, two lasers control performance of the thin film transistor and show that you can create metastable level and do very complicated computations. Guys show encryption, guys show power effect. You can train this because it's biological dielectric layers sensitive to humidity, sensitive to electrolyte presence. You can train them and you can modify logic and do this. Guys made 30 array transistors to control this and see what's happening there. This continued project.
You're doing your materials work. Are you doing it all on dry surfaces? Sorry.
So you're talking about your different materials and electronics. I wondered, are you doing most of this on dry surfaces or are you doing some of them in aquatic or wet surface?
You start, it's always wet chemistry assembly. You have this needle, so you have nanofibers as well. You start from wet state suspension. Key point is how processing will be affecting. We want them to do orientation to create films. We ending up in the solid films. They can be composite if you add them jelly stuff, they will be jelly. See if you control flexibility. And like for transistors, yes, of course, in the end, you have to print because then you use photolithography, use representative to build up. So we start with suspension and we ending up with a device that is basically solid state.
I'm tracking the solid state part. Are they functional in a wet environment? That's what I'm trying to
Yes,
ask.
they can
Okay.
do this. Biophotonic transistor, we study how humidity affecting. You can put them at different humidities, how it's affecting, because cellular structure, they're sensitive to humidity, changing light as well, that can be passing through. And we use this, we show that humidity control adding up flexibility. You're doing this.
Anything else?
How do you see the development of biophotonic transistors in the next 5 to 10 years as this unfolds?
It's in my review I did last year. So it's probably the way to do lightweight, super strong materials, not exactly right, because people who need lightweight or super strong material, they usually use them at extreme environments. Too high temperature, vacuum, you just kill them and you cannot get what you want. But if you're talking about things like sensors, human sensors, wearable devices, optical computation, in this case, you don't need to care about this. You're just adding up additional functionality. Like what I show you, sensors, they're compatible, they're biodegradable, we can put on humans without any effects, rush and everything else, and we can use them for a while. This kind of, I think, direction before, like I started doing this maybe 15 years ago. At that time, goal was for my Farzaneh, make them strong, lightweight, super strong, everything will be great. Yeah, and we made them. Excellent elastic modulized 150 gigabytes. Problem is that they're good at ambient conditions. And Air Force need them a little bit different conditions. It's basically after 10 years, we kind of move out from the mechanically structure-related components, we move out to functional components, electronics, photonics. I think it's kind of direction that bio-derived materials, so probably will be much more valuable.
Anything else? Yep. All right, thank you very much. We're gonna try again.
All right, everybody online, we have one more presenter. And we think it's going to work this time. But give us one more minute.
Well, thank you for the opportunity to present. I have a different kind of, turn it up. Testing? Or testing? I have a different kind of presentation. It's short on data and description of my lab and more focused on, hold on. Make this larger. It's full screen on my screen. I don't know.
It's fine. It's full screen.
We also have it here too.
It's full screen on the Teams. All right. Oh, it's okay. A little better.
Hold on, thank you.
All right. So I am the Georgia Tech Director of the Regenerative Engineering and Medicine Center. John Peroni at UGA, Ned Waller at Emory are my counterparts. REM, as we refer to it, is the remnant of the Georgia Tech ERC that preceded CMAT, that was called GTEC. That was the Tissue Engineering Technology Center that I think will be familiar. It's left quite a legacy in terms of the reputation in biomedical engineering and several and other departments, as well as colleagues that are in leadership positions that were junior faculty at that time. Many of you will also be familiar with CMAT. I'm not in CMAT leadership, but I am involved in CMAT. And as CMAT now approaches its transition from NSF funding, it's beginning to think about a transition plan and vision. We have other centers also that are at different stages of conception and execution. So I'm going to be talking to you a little bit about conversations that I've been in with leadership of the Cancer Technologies Center, the Immunoengineering Center, my own Center of Regenerative Engineering and Medicine, and CMAT. So my goal is really just to communicate a potential opportunity for a focus on engineering healthy aging. There is, which I'll speak about in just a second, some aging emphasis that exists at Georgia Tech that may be familiar with GTRI and may involve some GTRI investigators. But on the therapeutic standpoint, about how we can actually use engineering technologies, regenerative medicine and immunoengineering to engineer kind of lifelong wellness and resistance from disease, I don't think we're necessarily focusing in that area, although my argument is that we have some of the components. I'm going to fly through this because this is kind of thematic-centric, just giving you a sense of what the immune system is and does. It protects us from unpredictable injuries, infections, diseases, and unanticipated times and in different locations. And so it's kind of a what, when, and where problem and what it's supposed to do. And the way that it does this is through what we refer to as soluble effectors, mobile elements, and organized tissues. And so when I refer to things like cytokines and chemokines, these are kind of our soluble effectors, things that get produced by cells that exert changes in immune response. And so if I use words like immunomodulation, what I mean is we might hack into some of these existing signaling mechanisms to get immune cells to be in a certain state of activation, to be recruited into different tissues at different times in ways that could be desirable from a therapeutic perspective. And we have mobile elements, that is to say, we make cells that survey our tissues and our blood for threats of infection or potential tumor growth. And we have organized tissues like lymph nodes and lymph organs that can process signals. Most of us, I hope, are vaccinated. That whole process of getting B cells to make antibody that protects us from future COVID infections, that happens in kind of organized tissues. And so from an immunoengineering standpoint, we try to think about the ways in which we could create microenvironments that might mimic some of the locations where a lot of this immune response is happening, and to make more antibody or to engineer cells that can specifically go after potential tumor antigens. And there's a whole host of other types of immune cells that people are thinking about engineering. You've probably heard of CAR T immunotherapies. It's a central part of our CMAP ERC. You could also think about some of the stroma elements here, like the mesenchymal stem cells you may have heard of. There's also a focus of both the Marcus Center that we have here, that I probably should have mentioned separately, in addition to the CMAP ERC. And then there's a whole host of other cells that could be engineered in ways that could be utilized as a form of therapy or as a resource for creating bioreactors to make different types of those soluble effectors, right? So, we've been on this for a while. And particularly as it relates to CMAT and cancer immunotherapy. So, I just wanted to kind of give you that kind of backdrop, because I think that, CMAT is just one example, and immunoengineering is just one example of the ways in which we've already begun to think about how applied engineering technologies and GTRI strengths and things like automation could be leveraged in a large grant proposal like CMAT, in a way that Georgia Tech is recognized for research excellence and leadership, right? And so that's a story, So I think bioreactors is another thing, but so hopefully you appreciate what I just said. But now here's my argument. So here are the different, just the ones that and by the way, if I'm not including something in the list of existing centers and people groups, that's not intended. These are the ones that I know that I've had conversations about aging focus fairly recently, and things that I have some direct connection to. Okay, so as I mentioned, REM, tissue repair, regenerative medicine, biomaterials, immunoengineering, cancer technologies, and cell manufacturing. So part of what I really wanted to say and pitch to a degree, and if there's time, I'll give you kind of one example of how it played out in my own research, and why I think if we had some vehicle to create even more of a critical intellectual mass around aging as a problem focus. I think it could have capture a lot of synergy with people and projects that we already, and technologies that we already are working on. But, okay, so from REM standpoint, REM is thinking about its sustainability. So as an example, in our last REM retreat, we invited Todd Golding, who was recently recruited at Emory, and he was making these antibodies that could target muscle in ways that could help muscle grow. And one of the things that we know well about aging is that we lose the capacity to regenerate tissues and recover from injury. Muscle is one specific tissue that has a particular challenge in our ability to regenerate. And I'm interested in, making muscle grow. And so, the REM retreat was an opportunity to engage with him about, can we use your antibody in a model where, muscle is damaged in, an aging mouse model. And I'll show you an example of that in just a second. From an engineering standpoint, even just yesterday, by the way, Ankur Singh, I think, made a similar pitch to what I'm about to say about the way in which he's envisioning immunoengineering. Matter of fact, I'm going to just cut to the chase. So this is basically, encapsulate a lot of what we have been talking about. That is to say, I think that a focus on kind of collaborative solutions for healthy aging and immunity and regeneration just makes a lot of sense for us. I think that we have and are making significant advancements on the immunoengineering front, where we've got large grants and investigators that are working on organoid models, already thinking about ways in which those models could essentially replicate certain phenotypes associated with age, could already potentially be able to model some aspects that may be underlying mechanisms that lead to health disparities and could address questions of health equity. So to a certain extent, we're already doing that. We're already doing targeted therapeutic interventions to think about how we can address chronic inflammation at its source. We're doing metabolic profiling of tissues, both in health and in injury states. And we're looking at those things, in different kind of aging mouse models and thinking about therapeutic interventions. We're already thinking about health disparities research and models of kind of the biological heterogeneity of humans, and thinking about personalized medicine in a way that puts health equity as a point of real focus. And being here in Atlanta and leveraging potential partnerships with AUC, I think there are real opportunities to do that. And that, by the way, that was part of the pitch that investigators came to make to one of the Chan Zuckerberg biohubs that we applied for. We didn't get that very large $500 million grant in Chicago, and we didn't get it, is the point. But our idea really centered around our geographic location, the diversity of our population in the state, and our strengths to say, hey, we could be a leader in kind of this health equity engineering, and particularly as it relates to the immune system. And so part of what I think Ankur articulated, that we've talked about in REM, that we've talked about in the Immunoengineering Center, Susan Thomas is doing a great job at really spearheading a cancer technologies center. She's thinking a lot about resilience and resistance to cancer in ways that, she wouldn't be the first, but certainly we could be a leader in really thinking about cancer therapy from a prevention standpoint. And certainly, when we think about susceptibility to cancer, there's definitely an aging component to it. And so, anyway, so the idea is that we could, provided there were mechanisms to bring investigators together to leverage some of the research and technologies that they're pursuing, I think that we could really develop a leadership reputation in this idea of engineering kind of healthy community. I have a bunch of words here that I think are reiterating a lot of what I just said. And since our presentations are supposed to be short, I'm just going to maybe just skip to one kind of example of what kind of got me thinking about this from my kind of own experience. And I'll try to make it quick, but we think a lot about bioactive lipids. These are things that, among other things, can control immune cells, where they go, what states they adopt. And I mentioned a collaborator just a moment ago in regards to antibodies and how antibodies could be engineered in a way that could be used as a form of therapy. So there's a lot of information on this slide. I just want you to take away one thing. We needed an antibody that could fuse this thing called the ApoN domain, that is one of the domains on the HDL particles. So those of you who've done blood work and you're familiar with HDL, LDL, triglycerides, this is an HDL particle that we measure in the blood. This ApoN is a carrier or chaperone protein for this molecule that exists endogenously called S1P. When that molecule is presented on the ApoN domain, it tends to be pretty potently anti-inflammatory. We thought that we can exploit that therapeutically, but S1P, the molecule itself, is fairly ineffective in just systemic delivery. We needed to fuse it with an antibody. This is the Fc domain of an antibody, and so we wanted to make this long-lived circulating ApoN presenting antibody that we did in that we worked on in collaboration with others. And so then we needed a way to kind of monitor in real time how the antibody was actually working. And so we had this mouse model that we create a defect here in the quadriceps muscle of this mouse. And that defect essentially won't heal. It will fibrose, you'll have fatty infiltration, and you'll lose function in the muscle, much like actually happens in after human injury. But the challenge that we had is we really needed a way to monitor the changes in the properties of that tissue without having to sacrifice the animal. So we reached out to Stas, who was pioneering this effort to use ultrasound elastography, where he actually uses this acoustic radiation force to kind of probe into the tissue, and then he measures the shear wave propagation in the muscle, and to extract the muscle's mechanical properties over time after injury. And what we really want is we want to be able to mitigate the stiffening and the fibrosis in the muscle over time. And so what Stas was able to do for us, essentially, is over the course of about 28 days, what you can see here is that the muscle is actually increasing its stiffness after injury significantly without any form of intervention. And then when we use the antibody that was presenting S1P and mitigating inflammation, and essentially this is that immunomodulation idea that I talked about earlier, we can reduce that fibrosis pretty significantly and recover a lot of the function. And then that turned into a grant, but then we also, because I was unfamiliar with ultrasound and some of the potential that it brought to bear on the problem that we were interested in, we viewed this as an immunoengineering challenge. And one of the things that we wanted to do was to be able to monitor the infiltration of immune cells and the polarization states that they adopt once they got there, because that was one of the key mechanisms of fibrosis in the tissue. And so, to make a long story short, we reached out to Costas Arvanitis, who was loading cells with microbubbles, and he's actually using the cells themselves, the immune cells themselves, as contrast agents, so that you can monitor their infiltration into different tissue compartments, which became kind of another window of collaboration. And the way the story basically ends is that somewhat serendipitously, we left a cohort of animals in the vivarium unknowingly for about a year and a half. I'm very embarrassed by that, but that presented an opportunity for us to really address one of the challenges, which is we lose regenerative capacity the older we get. And so the question became, can we use our localized immunotherapy in a way that is more selectively and robustly effective in aging populations, right? And so the answer is yes, and guess what? That's the one slide that actually didn't come through my conversion. But you know, the answer essentially became the latest of one of our grant submissions, where we're starting to do kind of this aging-centric research. But the reason I kind of told you that story is that kind of all along the way, all of us kind of felt like we should be doing aging research. Aging should really be a focus of the research that we're doing, but we don't really have the ecosystem isn't structured in a way that centers aging in a way that that could help us, you know, advance the project. Anyway, so I think if we were to have more of a, you know, a focus on healthy aging as an idea, I think that there are many of these types of opportunities, many of these types of collaborations could happen, and I do envision that there would be a significant role for engineering technology in GTRI. Okay, so I'll stop there. So thank you.
That was very cool. Yes. So I had a question, maybe thinking but less about aging, but more about injury. So your tissue engineering, could that be applied to general wound healing?
Yes.
And then I was thinking with the monitoring of the muscles and how they heal, could that be applied to, even if I injure myself, could you tell things about my muscles healing or help them improve?
Yeah. So, for example, the technology I think was that Stas is developing. I think he first reached out to Andres Garcia. I think they have an R1 now where they're actually looking at the diaphragm and using this type of intervention to look at the engraftment and the therapeutic success of transplanted muscle stem cells into the diaphragm. So, and that's not an aging-centric project either.
Yeah.
But certainly it speaks to the idea that you can use this method as a way of monitoring kind of the therapeutic success of cell therapies, potential small molecule, or, you know, chemokine or growth factor therapies as well. So, and that's where we're going. I mean, we're doing a lot of that. But part of what I didn't communicate is that, you know, if you look at even, you know, PD-L1 immunotherapies, if you look at growth factor-based or cytokine therapies, or even cellular therapies, all of those problems are really more challenging in an aging population. So, you know, when you think about wound healing, it's much more challenging in an aging population. And we don't tend to think about that until relatively late stages in our kind of technology and therapeutic development. And what I'm saying is I think we should start thinking about it much sooner and develop models that can represent the aged state, both in vitro models and in our animal models, and develop our therapeutics accordingly. And I think that might open significant avenues for existing collaborations to extend their work into the space in a way that gives Georgia Tech a real leadership.
Yeah. And I was thinking people with certain conditions or disabilities, maybe monitoring, but also I would imagine for GTRI, I'm not in it anymore, but y'all can tell me, soldiers and wound healing is a big thing, I would think, and monitoring their abilities after injury. That was one of the things I was thinking.
Doug, that's a great point. And also one of the peer, I was a participant on a stakeholders meeting for a new arthritis initiative. And their first call from the peer review orthopedic research, through the peer review orthopedic research program, was to look at, it wasn't aging, but it was sex-specific differences after orthopedic injury for wounded soldiers. And some of the same things that we just kind of talked about how they're just, we need to represent different sex-specific phenotypes had so many parallels to, you know, the ideas that we've been talking about in terms of, you know, the aging effects as well. But, you know, I think for the opportunities for DoD collaboration, I think are.
Anyone else have a question?
Thank you. Agree on the DoD applications, and I've used mic too much, so somebody else please take the mic. But you want to ask about the mechanical waves of the muscle issue. I know that was a collaborator you provided that analysis, but was it only the shear wave, and were you only able to get the shear wave, or do they both exist in that type of tissue? So was this, you said it was specifically just the shear wave?
So in terms of how we measure the modulus in the tissue.
That's good too. Yeah. How was it measured? That's
yes. So I'm sorry I wasn't clear. The point is that using ultrasound, you apply kind of the ultrasound pulse, and then that shear wave propagates, and the speed of its propagation is related to the modulus. And so we're backing out, and that's what essentially, you know, what we're graphing here. We're graphing essentially the shear modulus of the tissue and its change over time. So this is at, you know, time 0 when the tissue is initially injured. And you can, what you're seeing in this increase is the stiffening in the muscle, right? Measured by the shear propagation and the estimation and the calculation of the modulus.
So kind of circling back to where these talks started today.
Mhm.
You're talking a lot about aging. Earlier talk was talking a lot about neurocognitive issues, motor related issues. Has there been any point where you're connecting the aging part of your interests over into the neuro part? Or are you mostly focused on more muscular autoimmune type applications?
Right. So has there been any talk of therapeutic development for diseases of neurodegenerative diseases and their presentation in an aging population? The answer is yes, certainly. It's not something that I personally work on, but part of the point I'm trying to make is that in many cases, the mechanisms that lead to dysregulations in the immune responses become or contribute to disease pathologies. Certainly that is true in the brain as well. And so should we understand from an engineering model standpoint what those immune dysregulations are, we're already developing some of the therapeutic approaches that can ideally re-educate immune response in ways that will be therapeutic. I'll give you an example from my own research. So we do some research in sickle cell disease as an example. One of the manifestations of sickle cell disease is a learning and cognitive developmental challenge that gets underdiagnosed. A colleague from Emory at the time approached me to do metabolic profiling and immune cell profiling in the brain of sickle. We had a humanized sickle mouse model that, you know, essentially asking whether social stresses can actually lead to neuroinflammation in sickle cell disease that was contributing to the cognitive and learning deficit. You know, and to make a long story short, yes, it was. There were mechanisms of inflammation in the brain that we were very familiar with in completely different contexts that were contributing to this neurobehavioral and learning deficit that we could then intervene in by conceiving of an immunotherapy that could apply, that was really targeting the brain. So those conversations are happening. I think that there are more opportunities to do that going forward. I didn't, like I said, I tried to do a disclaimer. I didn't include the neuro center group in my slide just because I don't have any connection to them. So this was very Botchway lab centric, I suppose.
Excellent presentation.
Thank you.
Really great to see this and see the whole, going from GTech to CMAT and beyond. Love it. I wanted to just, and this is not my idea.
Yeah.
But yesterday, Encore presented to the External Advisory Board, IBB External. Were you there?
Yeah, I took a slide.
Okay. So the comment that Vincent Ling made was a really good one. So thinking down the line, clinical trials are going to be really hard for any kind of anti-aging therapeutic. So I really liked his thought about thinking about veterinary medicine. Also, when you think about where pharma is right now, it's really, really difficult. And I thought that his comment about an initial indication or market, kind of beachhead that you could think about is animal medicine, getting clinical information from, and then moving on to humans. So I thought that was really, especially how we love dogs in this country. I thought that was a really interesting comment that he made.
Absolutely. And so John Peroni, who is a orthopedic surgeon, he's an orthopedic surgeon to animals. I don't know how you refer it, veterinary medicine orthopedic surgeon. He has made that exact comment that there are significant opportunities for us to consider these veterinary problems as the focus of our initial clinical trials and therapeutic development. Definitely. But I'll give you a very practical example. In REM, we funded one of John's proposals. So imagine trying to do a study in pigs, that, can get very, very large on, a $50,000, seed grant budget that is split between 2 universities. So that's challenging to do. And those type of challenges, I think, really limit 2 things. I mean, A, they just limit our ability to really reach the goals of that grant mechanism, which is to provide you seed funding that then you can go on and compete for a much larger grant, right? And if you have to reduce the scale and scope of those preliminary studies to such a degree that they're no longer competitive, that's a problem. But part of what I'm arguing as well is that, I think relatively speaking to a lot of our peer group, we don't have anywhere near the same type of reputation and presence in aging research. And I think it's somewhat difficult for us to bridge that chasm, even though we have the expertise and some of the technological, that our ideas, I think, are very relevant to these type of challenges and problems associated with aging. We're limited in our ability to really get there because we don't have a rainmaker who's already doing aging therapeutic development who's already here. And we don't have the mechanisms in place to provide those collaborative opportunities for us to get together and get the seed grant to apply for the aging research. And so I think if we really want to have a reputation in this type of research area, we need something that's going to move the needle. And right now, I think it aligns a lot with our strengths. I think it aligns a lot with what our existing centers are already thinking. And I think it's an area where I don't see us as being so far behind our whoever you would consider our peers as to not assume a leadership position in the near future.
Understanding that aging is a process and people are unique. And given that I represent a lab that does a lot of data analytics and has a healthcare division, what have you looked at? Maybe there's a lot of talk here about the experimental aspects and the work up to different animal models, porcine to human, that whole pathway, and pharma picking this up and what that transition path looks like. What about the data side of it for humans to be unique and the analysis of individuality of aging, like personal healthcare journeys and trajectories
unique biomarker footprints, or literally, I guess, a path of biomarker footprints through space and time. What level of interaction are you having on campus or elsewise with in that space of just the individuality of the humans and their data?
Thank you for that question. I really wanted to try to make this point. The data challenges that we have are significant. One of the biggest challenges we have is like data integration. I do a lot of lipidomics. Lipidomics has a bit of a challenge because we currently haven't gotten down to single cell level, like some of the kind of transcriptomic profiles that we can do. But we're looking at essentially metabolomic and lipidomic profiling. We're looking at transcriptomic and genetic profiling. We're looking at epigenetic changes as well. And we're trying to integrate the kind of data sets that we could generate in a way that gives us ideas about, potential targets and biomarkers. I can say that one of the things that happens in the model that I showed you, the volumetric muscle loss model, the injury model, is that you get an accumulation of senescent cells. Guess where else you see an accumulation of senescent cells? In aging, right? But the point is, what we're trying to figure out is, can we identify potential biomarkers of cellular senescence that can be targetable in ways that can eliminate those cells and essentially let the endogenous repair program take its course? But for us right now, especially when the data sets get really large, it's just a challenge for us to identify what the most promising targets and markers may be from the data we're using, much less trying to understand what its function might be when we have different types of data sets ranging from metabolomic to transcriptomic to epigenetic. So yes, we have a lot of challenges there. And if there are ways in which we can employ computational models or get just better at analyzing our data, particularly where we're attempting to integrate it, that would be great.
I want to make sure we have time for people to talk to each other after this, but I know, Ryan, we were going to have you say something. What were we going to have you wrap up with? Thank you.
Rebecca provided the call. There's a call PDF that.
Yeah, that's what you meant already.
Are we getting the PDF shared? I think this one's working. For those in the room, there's copies here, hard copies, if you'd like one, but I think Trina's pulling up one for the screen as well for those online.
Yeah, so there's a call for proposals. If you'd like to partner with GTRI on something, we can help you find somebody. And here is the call, so you know a little bit more about it, but you're not going to have to turn anything in until spring. So we have a while, but go ahead.
Yeah, so to give an idea about this call, it's IRAD independent research and development through GTRI. The focus of IRAD funding needs to have a DoD potential use case in the future, or bring some benefit to the DoD community, because that is the source of the funding. So be thinking about that when you're coming up with your ideas. The DoD has a lot of needs. So, that's a pretty broad window to look through things. We're at October 30th today. In November, we'll have another collaboration meeting. The proposals will be due in February. We can provide you with the proposal template. It's not a long process. We've tried to make it fairly simple. Yeah, it's not anything like NIH. A couple pages asking for, a description of the idea and the impact that it could have. For those on campus side, you do need to find a partner on the GTRI side to propose to this. And we can help facilitate finding that. If you have an idea and you're not sure, who to contact on the GTRI side, you can reach out to Leanne or anyone else on the committee, and we can help try to
facilitate those connections. We have example research areas, but it is really, a pretty broad lens. Some big areas: sensing technologies, biocybersecurity, biointelligence, distributed biosurveillance, modeling. It kind of runs the gamut, and we've had projects in a wide range of areas. So we look forward to seeing all of the proposals come in, and feel free to reach out if you have any questions.
One comment, it's not going to be in November, it's going to be in December, the next.
The next one of these is going to be in December. And we encourage you to come and listen to other colleagues talk about what they do to help create those synergies. Yes.
It'd be great if we would list some
of the proposals that got funded in the past, so people have an idea of the types of things that we're looking for.
We are doing that. So we are getting ready to set up a web page. I think Brian has helped looking into that now, where we're going to have previously funded projects listed so that you can get an idea of some of the things that have been of interest, that have come out of these discussions previously.
You probably anticipate what I'm going to ask. A lot of times when I see a call like this, I'm looking at the example areas, and it seems somewhat exclusive of the kinds of things that we do in like regenerative medicine. And so when you think about battlefield orthopedics or things like, implant or, surgical infection problems, I did see microbial sensors that, maybe touch on those. But I guess my broader point is, sometimes the research areas to me don't necessarily encompass things that I think I do. But if there's a broader opportunity, then
Some of these are actually a little more past or ongoing areas, so kind of extant things. We are open.
Okay.
To more. Yeah.
Absolutely. We're open to all kind of new ideas in this realm.
Yeah, so Ed and Farzaneh particularly, finding a partner in GTRI is the first step. And I'm not sure the best way to facilitate that because
Is it like consider list of at GTR and check the research, obviously we would do
that's the difficulty.
So sometimes what we do is we have GTRI people talk about their groups and what types of different things they do as well at these meetings. But I think that typically what I do if I'm looking for a partner, even beyond this effort, is I'll start with the chief scientists in each lab, which we have a few of them here and we know them all. And that will help. They usually are really familiar with the different types of research going on in their labs to be able to pick that person for you. But I'm going to hand it off to a GTRI person to also answer that.
Sure, I can give some for the Advanced Concepts Laboratory in GTRI in the biospace, or like this IBE research portfolio space. There's a lot of things that go on in ACL, but we'll focus on the IBE space. There's sensor technologies being developed for infectious diseases. There are biocybersecurity projects that we've been working on. So one example is like looking at DNA as a cyber attack vector. There's biointelligence work going on, building predictive models or classification models to source materials. There are distributed biosurveillance. So we lead the Georgia Pathogen Genomics Center of Excellence, using genomics and trying to support public health and epidemiology in that way. So there's a large range of work going on, but I think, and I'll pass it on to others to talk about their work as well, but the best way to get connected with someone in GTRI is really to reach out to the committee, because not all of our work is allowed to be published. So it's difficult sometimes to, especially when we're talking about like DoD portfolios.
Reach out to the
Like the
Trina, who helped organize this.
I'm sorry, I also don't know what's the people's names.
Yeah, so I'm Leanne West. I'm helping facilitate this interaction across campus. Cindy Sundell is on the academic side, in CMAT, works with Ed. Greg Gibson here does more things in like genetics, genomics, and analysis. Here we have Rebecca Hutchins, and so she's in GTRI and is one of the ones who does a lot of the research in the biospace in GTRI. You have Ryan and Nick over there, and then do you want to say which labs you're with?
So information, I guess we're still online.
Yeah.
Information and Communications Lab, serve as the chief scientist there. We do have a health division. This particular committee, though, is more about biology and engineering. But that doesn't mean that we would like to exclude. In fact, we'd like to include all kinds of health collaborations. We're very interested in that. We were talking about with Grishma, and I forget the last name, about healthcare trajectories recently. And so we have a health division. Very interested in that, health disparities and equity, and anyway, a whole range of things. What I'd like to do is to say that we definitely are interested in the healthcare space and working across labs on that. And I did pass, we have a chief scientist council within the lab, and I passed this meeting to the division heads. Each division, just to give a cross section of our labs, each division is about 30 or so research faculty members. So I think, you have the capture area of over about 150, just for the lab that I represent, about 150 research faculty. Now, they're not all in health, or some are going to be in wireless communications, but about 30 of them are in health, and the chief scientist for that division was on the meeting today. So we did use some capture of people across GTRI, and we'll definitely have follow-up conversations, and we'll be exchanging some emails with you. We have some work in edge intelligence and big data for healthcare. And so somewhere between like wireless communication among smart devices and people's personal health data and its privacy, back to all payers claims database sort of data, we have those competencies within our lab. So there's a lot to wrangle, but also a lot of opportunity for being able to span individuality to the big data problem.
Sensor technologies are a huge thing in GTRI too. So monitoring health or monitoring conditions, monitoring machines, like there's a lot of different things in there that could, yeah, so that could be a big part of it too.
Yeah, we break down what we do broadly across GTRI as sort of sensors, information, and systems. I mean, that's kind of bread and butter across GTRI. And yeah, just to second what everyone has said, there's upwards of 1,000, close to 2,000 research faculty at GTRI. Yeah, and yeah,
1,500 researchers faculty, not teaching.
And yeah, unfortunately, you can find almost none of them online. We don't have websites. Yeah.
Yes, multiple buildings.
Yeah, so at Georgia Tech campus, they do have a few sites off campus where they have a few faculty researchers, but for the most part, all of those research faculty are at GTRI on Georgia Tech campus.
And they also.
They do,
Yeah,
so they're
everyone
considered faculty at Georgia Tech.
Yeah, yeah,
It's like a college, but it's out, it's outside of the college.
Part of why we're doing this is we've had limited connectivity in the past with campus. Yeah, a lot of what we do is DoD and can't publish it or talk about it. So
you can find us.
Oh,
yeah, you're how long have you been at Georgia Tech? So.
I mean, we're in the directory. We can
Yeah,
find
we are
that,
in the directory.
but you can't easily find what people's specialties are or particular research.
We can help you do that. Like, that's kind of why we're here, is help facilitate that interaction. Yeah. Did you have a question?
Yeah.
No, very quickly, since we're running down, I'm Nick Geis. I'm from the Cypher Lab at GTRI. That's nominally our cybersecurity lab. So we're involved with Rebecca's lab and the biocybersecurity effort. I guess that just makes the point that some of our research cuts across labs. It to some extent doesn't matter for you all which lab somebody is in, which is why we always advise, just talk to us and we'll try to connect with the right person. Despite being the cybersecurity lab, my lab does a lot with materials. We have people that work on graphene, biosensors, especially chip-based biosensors, and then also, you know, molecular data storage and sort of the informatics side of biology.
Okay, I have one question or comment. I did some collaboration with GTRI. I know how it's difficult. Usually it works like this, is my advice. Yes, send some suggestions to link, for example, title, maybe a couple lines, and they will give you contact point. This is how I initiated a couple of collaborations. Yes, I have specific questions. So do you think about multi-PI projects, and what about students? I know that at some point was limitation only US domestic students and some restrictions on how I handle this in my lab. What about this? It's kind of open. Can you comment on this? What should I think when I design this?
It can be multi-PI for sure. I think there are still limitations, but I'm going to let a GTRI person say.
We'll take turns on answering the question. There are different parts of that, depends on the source of the funding. So today we're talking about our independent research and development funding. That's not, it's derived from government contracting, but it's not subject to those contract requirements. So we can do fundamental research under that funding. And so then we issue a statement of, you know, research abstract to the academic side of campus. We, you know, we have now protected that for the professors. We know you need to publish, graduate the students, right? So we,
but it was a major problem.
it has been in the past, actually. So we've worked on that process now where we, you know, it's a one pager and we declare what will be done that is fundamental research and that protects us and protects you for the ability to publish. So you're not subject to any NDA or anything like that. And so we do that under our IRAD, Independent Research and Development, IRAD funding source. But we can both employ students as GTRI employees, particularly as easy as a US citizen, that sort of thing, for full access to facilities and all that sort of thing. We can also fund the students within the professors' labs, and they don't need to be a US citizen for that if we declare the fundamental.
Good to know. I had difficulty when they try to kind of close my lab, make a separate server, make this room closed.
It's too much. Sometimes it doesn't make sense. Sometimes we're willing to try something that maybe is logistically problematic.
Good thing you think about.
Let me just add one thing. Yeah, so a US person really makes it easier for some of the, yeah, but citizen not necessarily, and really neither is required. With there are ways to work around it, but yeah. The one thing that we do ask all our collaborators, faculty and students to agree to is with this IRAD funding, we do have a sort of internal review on publications before, you know, conference presentations or journals, if it's derived from the IRAD funded project. That's not a technical review. It's just, you know, GTRI wants to look at it and make sure that, you know, if our name is on it, we're aware.
We've recently been thanked for something where we said, wait, did we fund that? You know, so it can be challenging.
And then we've also started asking for sort of like another funding document that IPU would submit to NSF. Again, just to see if there's any, you know, sponsor conflict with work. But so those are the two sort of strings attached that I'm aware of.
You told that you're going to provide us with template. What, give it like a suggestion scale? NSF type projects like 25 pages, like white paper to DoD, like 3 pages.
5 pages.
So it's more like white paper to DoD.
Yeah.
Okay.
Yeah.
Okay.
Trying to keep it easy.
Okay.
Then is it like preliminary data, or is it more like, you know, they are looking for high risk projects? What kind of?
So for this effort, we are really looking for the high risk projects if we can. But I think that, for example, one of the ones that got funded in this last go around was wearable sensor technology, but it was bigger than just building one sensor to look at something. It was really, how do you solidly integrate it from kind of head to toe with what you can determine about the health and well-being of the soldier, like while in action. So it's
And
bigger.
your published data may be high risk to us, so we shift the risk sometimes. You may have something that's very solid, but then it's hard for us to adopt. So there's the risk can be managed one way or the other. Sometimes we prefer, you take more risks, sometimes we prefer.
So you're kind of acting like FRL. Yes, they're also like splitting the fundamental, whatever they're doing for themselves, but they kind of handle it.
Yeah. In this effort, we'd love to see something that was really kind of different and groundbreaking. And, you know, if you see it in your research that this could be a game changer with what I do, like what I hear from my colleagues in the field or the thought leaders. But we have a piece of the puzzle that I think if we did this and this and this, that we might be able to start tackling that. That's the kind of thing we would love to see. Like, where are the game changers?
So this is probably sounding like a really long list of GTRI people to reach out to, but specifically to what you're talking about and maybe a little bit with you, we do have a GTRI neuro group that is mostly just a repository for knowing what people in GTRI are interested in neuro applications. And we meet periodically and I have that list and I can actually be able to direct you that way. So talk to me. I'll give you a card. Just
Yeah.
And I would like
Did anyone else have a comment?
Yes, I think GTRI is we can grow into
yes.
as well, right? So this diagram here has cybersecurity, biointelligence theory, and computation, distributed biosurveillance, sensing systems. What you and Ed are talking about doesn't, at least to me, and the materials work doesn't necessarily fit into those three, but that doesn't mean that next isn't going to incorporate that. So you might want to think about getting 2 or 3 colleagues, Farzaneh, in the systems neuroscience realm, and then getting together and have, okay, the more you have, the more it dilutes the 300,000, but the more you have, the more likely it's to be a big award than a 100K award. So I'd be thinking big. Let's say with you, Ed, like in terms of aging and immunoaging, don't just make it an Ed plus $1 GTRI, but make it Ed plus Ankur and forbid plus Greg and put in a thematic thing, because what we're looking for is it's going to grow into an ARPA H, basically, or a DARPA.
Yeah.
Right. And that's not necessarily going to come out of a single investigative thing. You really want a systems thing.
For DoD, we can think, you know, that the soldier starts out enlisted or something, and then they go all the way through and they become veterans and VA and all that. There's this whole spectrum of.
So I was going to ask, what's the recommendation for the number of PIs? So you're saying it should be ideally, well, 1 GTRI for sure. Then let's say 1.
Well, it has to be one of each, please.
But you're saying maybe we can go like to 3. It doesn't have to.
Yeah.
Is there a Slack chat that we can use? Potentially.
So the question was, is there a Slack channel we can use to connect potential collaborators?
That actually has got security walls and stuff around it. It's hard to set
Yeah.
up GT Slack channel goes outside GT.
I think that's maybe a
Yeah.
tricky.
That would be a difficult.
Apparently within GTRI, we don't have an IBE specific one. We use the Chief Scientist Council. Yeah.
So we can try to figure something like that out, to figure out how to break the firewalls between the two, maybe. But also, again, I think, for example, if you and Tabitha hook up and she has her neuro people and you have some others on the academic side, let's get you all together talking, right? And you can come up with your own way to discuss things between people, for example. I mean, I think there are ways around it if we need to. We are building a web page, and that should be helpful. Trina sets everything up so she can send everything to me. If you want to reach out to her, she's probably the information that you have readily available, because she does send you all the emails for all of these things. And we'll just make sure to get you in touch with the right people.
Just out of curiosity, I mean, you're thinking about soldiers and battlefield orthopedics. That's one potential patient population where there are problems, but there's, of course, there are veterans and their care as well. There are problems, as just an example, I'm not necessarily pitching it, but, you know, as an example, there's, I know there's a lot of interest in somehow integrating rehabilitation sciences alongside, you know, things like regenerative medicine and, you know, more, you know, joint replacement or arthritis therapies. You know, and I guess question number one, the easiest question is, you know, are, you know, VA-centric problems also within scope? And you know, and I guess question 2 is, I guess I want to understand I'm in a lot of ARPA-H discussions, you know, about, you know,
we position ourselves to write ARPA H-like grants? Well, I mean, so one of the first, I think ARPA H's first call was like for osteoarthritis or something like that. Like, the Nitro ARPA H, the first non-open call, or the one where they solicited, they wanted new technologies for, joint replacement or something like that. But I guess my question is that for a problem like that, where, it is definitely within scope of something like an ARPA H, and dealing with, kind of arthritis and joint replacement has relevance for kind of military sciences and battlefield orthopedics as well. I'm still trying to kind of solve the puzzle of within our, what's the starting point to kind of do what Greg said in terms of identifying potential collaborators from within GTRI, because that problem still sounds to me to have quite a significant distance from what GTRI currently focuses on. But so I don't know how to ask this best, but like, how do we really, how do I judge what kind of next level new areas would be of potential interest to GTRI in ways that aren't necessarily directly connected to what GTRI is currently doing, right? Because I'm not in GTRI, so it's a little hard for me to know like what future areas would be of interest if it's not already reflected.
Well, you have to put an application and to get feedback.
Email.
So,
Email.
Yeah.
So, and I think a lot of times GTRI too, they focus on their capabilities,
Yeah.
so it's getting a line of sight into those capabilities and how they could be applicable too.
Not just slacking on your behalf right now.
Okay, all right. And can I ask one quick unrelated, but somewhat unrelated question? This could be, what I have to ask. I'm also the director of the Engages program in the SURE program, and I don't know to what extent GTRI investigators are interested in, or when we talk about students, whether you might be interested in kind of K-12 outreach connections and undergraduate research opportunities in the summer and those kinds of things.
Yeah.
I'd love to follow up with someone if there's.
They have a whole program dedicated to K-12 outreach.
Okay.
STEM and GTRI.
Yeah.
But can we connect to it? Okay.
For sure. So I do that.
Good question. So the PIs need to be like all Georgia Tech affiliates or no? Does it have to be? So like anyone?
I think for the IRAD money, it has to be Georgia Tech affiliates.
Doesn't have to be,
It doesn't have to be.
but
Yeah, like it doesn't have to be, but the purpose of the IRAD portfolio is to build capabilities and knowledge and technology within GTRI and Georgia Tech that we can then leverage into future sponsored programs. So while there's not a strict rule against it, we would ask a question to say, explain to me how sending this funding to someplace else. Right?
Well, Emory counts as out or does Emory count as in?
Emory is easier to justify.
Emory is easier to justify, and a lot of the people that we work with at Emory have joint appointments at Georgia Tech, so that is easier.
Okay.
What about UGA, large animals, big dog, clinical studies and, larger animals, is that?
I think it's something, I think we can consider it if it's something we can't do in-house. I mean, probably the PIs should be Georgia Tech, but we can have collaborators and we can send a portion of the project outside. There is a way to do that.
Anyone else?
All right. I just want to thank you all for coming today, being great participants, asking a lot of really good questions about how to make this all work, but also giving some really awesome ideas about different areas of research. I think this was absolutely fabulous. So thank you so much for participating today. And again, reach out to any of us and we will help make these things happen. Thank you.